Hi, my name is Miller Tran. I'm a co-founder and director of research and development for Triton Algae Innovations. And so Triton is a company that is dedicated to positively disrupting the food ecosystem. So really what that means is developing processes that are more efficient so that we can actually feed a growing population, uh, providing food that's more nutritious than our current nutritional profile, and also providing compounds that fill in nutritional gaps inside our current food chain. And so at Triton, one of the things that we constantly think about is how food is grown now. And so if you look at plants, one of the biggest gains that we've had in growing plants was applying fertilizer to the ground. And so this happened in the late 60s and in the 1970s. However, a majority of the nutrients that we put into the ground don't actually end up in the plant. And so if you look at the picture on the left, what you have is a graph of nitrogen. So for the amount of nitrogen that actually goes into the ground, only a little bit more than 60% ends up in the plant. The rest of it ends up running down the rivers, the streams, and ending up in the ocean. But what's more worrisome is phosphorus. So phosphorus is mined, and so there's a limiting amount of phosphorus that's actually available to grow crops. And so of the phosphorus that we put into the ground, only about 20% of that makes it into the plant. And so really, if we are intent on actually feeding a growing population, a population that's estimated to reach about 9 billion in the year 2040, we have to develop processes that are more efficient at using these fertilizing elements. And so at Triton, that's what we do. We actually grow a little green algae called Chlamydomonas reinhardti. And the way that we grow this algae is to grow it in stainless steel fermenters. And so why do we do that? And the reason that we do that is that we can control the nutrients going in, and a majority of the nutrients that go into the process actually end up in our algae. So greater than 90% of the nitrogen and greater than 99% of the phosphorus actually will end up in the algae. Whatever is remaining can actually be recycled into the following run. So a much more efficient process than actually growing plants out on arable land. And so what does that look like? So if you look at the picture here on the top left, what you see are the different types of algae that Triton can grow. So although we work with a specific species, Chlamydomonas reinhardti, there's different versions of that species, and you really have to identify which of these species can grow under heterotrophic conditions in a fermenter. And so when we first got this strain of algae and we tried to ferment it, it didn't really actually grow very well. And after a few months of optimization, we were able to increase the productivity and the titers of our algae to about 30 grams per liter. And now we can actually grow our algae to greater than 100 grams per liter, making this process much more economically viable. And at a company, one of the things that you have to do is not just bench science, you actually have to validate your process. And so we've worked with contract manufacturers to actually scale our process up to about 3,000 liters 3,000 liter tanks. So what does that mean for production costs? That means that our estimated production costs based on tech economics, we could actually produce our algae for about $2.17 to $7.76 per kilogram. And that just really depends on which contract manufacturer you're working with and how large your scale actually is. And so that's great. And one of the exciting things for us is when you think about product development, you can't just have one color. So oftentimes when we go to people to manufacture different types of finished products, they kind of give us this kind of look when they look at the green, because the green doesn't mix into a lot of finished products. And so what we've developed at Triton is a process by which our algae will not turn green, basically not produce chlorophyll, so that we have a yellow, naturally occurring strain of algae that we can incorporate into many different products. So one of the things that we wanted to see was not only is our algae a new source of food, but was it actually important for improving the gut health of humans. And so the way that we did this was we actually tested it in mice and pigs. So if you look at the image on the left there, we did a study with John Chang, a professor of gut immunology at UCSD. And in this study, we basically feed mice a compound, dextran sodium sulfate, and this compound mimics the effects of IBS in mice. And what it does is it causes the mice to get diarrhea and lose a lot of weight. And so that's the graph in the red. And so what you can do is you can test different dietary interventions to see how well those interventions actually help the mice recover their weight. And that's in the blue there. And so when we fed the mice our algae, what you see is that they recover from the detrimental effects of dextran sodium sulfate much quicker. And so that got us very excited. And so then we did a study down at Texas A&M with 118 piglets. And so we wanted to see if our algae could perform as well as antibiotics in these piglets. And so what we did was we had pigs that were fed their normal pig chow, and we measured the instances of diarrhea. And then we also had pigs that were fed our algae at 2 different percentages. And then we had pigs that were fed zinc oxide with their normal diet, and also oromycin, a common antibiotic used on farms, as a part of their diet. And what you see is that the pigs that actually receive our algae get decreased instances of diarrhea similar to pigs that receive zinc oxide or oromycin. And so very exciting data. And so now what we're doing is we're testing our algae in a dietary intervention study up at UCSD with Steve Mayfield's lab and Rob Knight to see how well our algae performs in improving negative side effects associated with IBS and also how it impacts the microbiome. And so that data is forthcoming, but very exciting times. But one of the things that we talked about is actually producing something that is more nutritious for people who eat our algae. And so in the 1960s and the 1970s, we developed a lot of seeds and a lot of cereal crops that are very full of carbohydrates or fats. And unfortunately, in a study done in 2015 by a group at Harvard, what they showed is that we just don't eat enough protein. And so what's nice about the algae that we use, Chlamydomonas reinhardti, is that when you grow this biomass up in a fermenter, it ends up being about 48% protein. So it really fills that nutritional gap that we're talking about that we really need to fill in the next 20 years. Okay, so that's one part of our business. The other part of the business that Triton does is we use synthetic biology to produce unique compounds that are important for nutritional health. And so to do this, we had to develop a whole set of genetic tools that were amenable to heterotrophic growth conditions. And so the genetic tools that you use have to match up with the processes that you develop. And so since a lot of the tools that were developed for Chlamydomonas reinhardti were developed to work under photosynthetic conditions, we had to develop a whole suite of new tools. And so we did this, and we examined now over a few thousand different vectors, and we identified some that were capable of accumulating proteins to a large quantity inside our algae in heterotrophic fermentative conditions. And so then we decided, okay, well, what are the nutritional gaps that we're going to try to fill? So as it turns out, one of the areas of nutrition that is very well studied is infant formula and breast milk. And what you see is that in infant formula, because it's based on cow's milk and not human's milk, there are a lot of bioactive proteins that are actually missing. And this could be osteoponin, lactate soluble CD14, lysozyme, lactoperoxidase, or many others actually. And so a lot of the infant formula companies to this day are seeking out sources of these bioactive proteins to add back into their infant formula. And this has been seen with lactoferrin and also MFGM. And so the one that Triton focuses on is actually osteoponin. And so osteoponin is a protein that has multiple effects, and the primary one is it improves your immunity against a lot of infections, and it also, from what we can tell, also improves cognitive function. So one of the things about osteoponin, though, is that it's heavily phosphorylated, and so the protein expression that needs to be used to actually make this protein has to be able to phosphorylate the protein. So on the left, what you see is the native sequence of osteoponin with the different phosphorylation sites. And so when you try to actually make osteoponin in E. coli or you try to make it in yeast, you end up with a protein that is actually not functional. And so at Triton, one of the things that we stumbled upon is when we produce osteoponin in the chloroplast of our green algae, what we see is that we end up with a final product that is actually phosphorylated and fully bioactive. And this bioactivity has been confirmed in conjunction with a doctor of neonatal health up at the UC Davis, Bo Lonardall. And so, again, very exciting times for Triton. And our company is 100% dedicated to filling these nutritional gaps. And I think that's all I have to say today.